do that? Well, it does that by stimulating, using an action potential. It stimulates the cell in the posterior pituitary to release its hormone that has been stored. It just excites it to release it. So the point is, it doesn't have to be a nerve cell. It can be an endocrine cell. All right? It can be another type of tissue. For example, it can be a part of your liver. It can be a liver cell. It can be part of the gallbladder. So in other words, there are many different cells that will respond to an action potential and then do something upon being triggered to do so. All right? Certainly muscle cells can be post-synaptic cells. We're about to get into that in a minute. So what else do we need to understand then about this synapse? Well, this is really kind of important, so I hope you do get a handle on this by next week. An action potential moves down to the terminal end. So this is the terminal end of an axon. Right, so this is where it ends. Okay, remember, there are many of them. All right? So then, how do I then stimulate, how does the terminal end of an axon here stimulate a postsynaptic cell to do anything? All right? Well, basically, what you have to do, essentially, if you're going to create a synapse with a cleft in space, is you're going to have a molecule here that somehow is released from this cell, and then obviously is received by this cell, which then can elicit a response in the postsynaptic cell, whatever that may be, whatever that may be. All right? So what is one or more of the classic, what we call neurotransmitters that actually performs this function? Well, it would be acetylcholine. So I would expect to see a lot of acetylcholine at this region of the axon, its terminal end. Okay? And I know it's designed to be received by a postsynaptic cell. Well, but they actually are created within these clusters. All right? So you take these molecules and you actually put them in these vesicles. Believe it or not, when you go into your more advanced courses in physiology, we actually know, we have a rough idea about how many molecules can actually fit in a vesicle. I mean, you can actually study it. It's kind of cool. All right, you can't just put an infinite number of acetylcholine in these vesicles. They're designed only for something kind of cool. Okay, so you're gonna wrap these up. All right, and if the action potential comes down, the magnitude is strong enough. All right, it's going to cause calcium to infuse from here into the cytosol in this region. That then causes a series of chemical cascades, molecular events, which then, all right, encourage these to move from here to the cell surface. They then empty their contents. They rupture, empty their contents, in this case, the neurotransmitter, into the cleft. It's then received here. This reception then sets up another set of chemical cascades. Remember, we haven't ironed all of those out yet. We just know that, these pathways exist. And that then initiates a new action potential, which then causes this cell to respond in some way. For example, if this were a muscle cell, then potentially, if this were a bicep, then those cells would contract. That's simple. Okay? So those are the kinds of relationships that you need to understand, and those are some of the more fundamental sort of processes that I'm going to hold you responsible for. All right? And also understand, oftentimes if we're going to influence in some way a neural pathway, especially using a drug such as morphine or whatever the case may be, this is a great place to do it. In other words, inhibit reception. Because remember, that's what inhibitors do in a natural way. All right? So if you inhibit reception, then you inhibit responses. And in many cases, that's a useful thing to do, especially if you're under a lot of pain. So again, this is an easy target, these receptors. Or you target the neurotransmitter, inhibit it. In other words, bind to it, make it useless. And then it can't bind. All right? But now also remember that most of these molecules will be degraded. They have a shelf life. So they will be degraded, and sensations, for example, will be restored. So this is why this has to be heavily monitored. Okay, but again, try to understand some of the more practical applications of that as well. Okay, so that said, all right, I also want you to be familiar with, when we talk about neurotransmitters, it's a lot like hormones, right? Be familiar somewhat with their origin, okay? Because in some ways it's the same as being familiar with hormones and what their structural, in other words, what their basement structures would look like. If we say that a hormone has a peptide, polypeptide origin, you should have a sort of a visual of what that might be like. If it looks like it has as a primary structure a steroidal structure, you should sort of understand what that looks like, the lipids. You should sort of be able to relate to that. All right, but again here, I don't want you memorizing all this stuff. Okay, I don't. But certainly be familiar, all right, with more of what we look at, for example, dopamine or norepinephrine. In other words, what are they? Where do we see them with relationship to pathways, central nervous system, etc.? So please take a little bit of a look at that, all right, in terms of your study. Basically, we've been talking about ligand-gated. Ligands are chemical messengers. Acetylcholine is a ligand, all right? It causes something to happen, all right? Gated ion channels would respond to those ligands, okay? You just saw that in that reception at the synapse. So again, all right? And then also be familiar with your PTSD, your IPSD, all right? In other words, how it works. And also, again, ask yourself this question. If you want to generate an action potential, which one of these would you have to have in a higher concentration? If you want to generate an action potential, you're going to want to see more of these. If you want to slow down, or possibly even stop, in other words, don't allow the signal to move any further, what would you produce in high amounts? You would do this. All right? Basically, sort of understand the relationship. In other words, action potential, the induced of an action potential is associated with high levels of EPSP. Stopping action potential from forming are associated with high levels of IPSP. That's how it works. They're antagonists.